Good morning. Thanks for joining us bright and early on this beautiful Monday morning. My name is Scott Osborne. I'm Vice President of Economic and Corporate Engagement at The Ohio State University. In that capacity, I lead the Corporate Engagement Office, and that office includes Technology Commercialization and the Keenan Center for Entrepreneurship, which is the umbrella that we support startups from the university. I want to welcome you and thank you for coming and being a part of this second program to be part of Startup Inventor Showcases. And I want to particularly thank our speakers this morning for coming. You know, those of us who are involved in technology commercialization know that many inventors and would-be inventors don't really have a clue sometimes as to how to advance their ideas toward the marketplace. So that's really what this series is about, is to let you hear the voice of those who have gone before you to advance their inventions and move towards the marketplace. So I also want to extend a special welcome to those of you who are at Nationwide Children's Hospital this morning and listening in from across town. So without further ado, I want to wish you a good session today. I hope you'll come back in the months ahead. And again, very special thanks to our speakers. I'd now like to introduce Dr. Jan Weisenberg, who is the Senior Vice President of in the Office of Research. Jan, please.
Thanks, Scott. Good morning. Yes, I'm Jan Weisenberger. I'm from the Office of Research. I would also like to add my welcome this morning. We are so delighted that this series is underway. We think that our faculty are an incredibly creative and innovative group of individuals. We think our staff are similarly innovative and creative, and we're astonished at how innovative and creative our students are. This is a phenomenal place to work. So I'm going to go a different direction for a minute. This is our sesquicentennial year. You've probably heard that. It probably doesn't resonate with a lot of you, but one of the things I've gotten to do this year is a project going on in our office called 150 Years, 150 Innovators. And we are featuring faculty across the entire 150-year history of the university who have done amazing research breakthroughs, who have done amazing innovation. And it goes all the way back to our very first faculty. There were only 7 of them. They worked under incredibly difficult circumstances. There are stories about the physics professor having to borrow a pail and a rope and a pulley from the construction guy so that he could run the physics lab. I mean, it's just amazing. But our first faculty were intrepid scholars. In fact, the very first faculty member, Thomas Mendenhall, invented something called the Mendenhall gravimeter. And it was the most accurate timekeeping piece in the world at the time of its invention. So I think about this spirit of innovation that has been here at Ohio State across our entire history, and it continues today, and this series just shows one piece of the incredible creative spirit that our faculty have here. So thank you all for coming this morning, and I'm going to turn it over, I think, to our next speaker who's going to be doing some introductions.
I think is this hot that I could just use this? All right. What's that?
Just minimize it with, which one is
This one's me.
okay.
Yep. Excuse me. Yep, I'm just gonna get on the right side. Thank you. It's not on the first slide here. Where's our? It's about 5 in. Pulled up in a couple different places. Thank you. Hello, my name is Matt McFarland, and I am the Vice President for Commercialization at Nationwide Children's Hospital. I'm not going to take a lot of your time today. I want to get right to Dr. Breuer and his work, and I think we're going to have a lot of fun listening to what he does. It's our pleasure as the Office of Technology Commercialization at NCH to work with Chris. And so I won't take too much time between now and getting to Chris, but I do want to take a few minutes today to have the opportunity to share with you what the OTC is at Nationwide Children's Hospital, and to the extent you're not that familiar with NCH, give you an opportunity to understand the research enterprise at NCH a little bit better. So what I have up here to start with is a copy of, or a picture of our Journey to Best Outcomes, which is our strategic plan for the years 2017 through 2022. And the reason I bring it up here is not to spend a lot of time on it, but to point to the fact that the hospital system has recognized for a long time that to grow from a regionally good hospital to a nationwide, no pun intended, health leader and an international leader really in pediatric medicine, research enterprise is a really, really important part of that. And, you know, we've grown a research enterprise that's pretty significant over the last several years. We have close to 1,500 employees now. This is a little bit of an outdated slide. Working at the Research Institute, nearly 200 primary investigators, over 100 trainees, and that's organized into 13 different centers of emphasis. I won't go through all of them today, but some of our probably most commercially successful ones, which is the area that we work, are the Center for Gene Therapy, Tissue and Regenerative Medicine, Cell Biology, or cell therapy rather, and microbial pathogenesis. We have several cores to support the work that goes on, clinical manufacturing. You'll hear a little bit more later today about our regulatory group from Chris Schilling. And we have a robust clinical trials infrastructure. And that's all enabled a lot of research to happen. And over the last several years, we've actually seen the total amount of external support of our research at NCH climb rather rapidly. It doesn't show it on this particular graph, but the total research enterprise, because of money coming in from the hospital as well, in any given year right now, is right around $200 million a year. So it's a significant enterprise. The last time I checked, and this is also an outdated statistic, but probably still relevant, we had right around 50 R01s at any given time at NCH. And so if you compare that to OSU, and this is the wrong audience to get this number wrong in front of me here, but I think, you know, they're up in the mid 200s to even higher than that at R01s at a time, like maybe 300, somewhere in that range. So it's a significant research enterprise. It's not as big as what you see at OSU, but we like to say we're small but mighty in what we're able to accomplish. The other thing I want to point out here is if you look at the total amount of dollars coming from what we would call other, which often can be like commercialization forms of funding and industry funding, that number's grown significantly over the last few years. So I guess I'm saying all this to simply try and convince you that we do have a significant research enterprise at NCH. And the other thing that NCH takes very seriously, if you look at this slide, the title of our strategic plan, Journey to Best Outcomes, for us to have the best outcomes for kids, we have to translate this research. We have to get it to the kids. We have to get it to the clinic. And so that's where our Office of Tech Commercialization comes in. And when we think about the things that drive us and the reasons why we do what we do, we kind of put it into 5 categories. We think about engagement with industry. It's really important for faculty to have the venue to be able to see their translation spark relationships with industry sources of funding, industry sources of technology. It's naive to think that you're going to do cutting edge research in the world today and just depend upon federal funding and not have any interaction with the for-profit sector. Faculty retention. As the typical faculty member has evolved from maybe the 70s or the 80s or 90s into a world now where, like I said, this industry is a part of what they work in and what they do. They want to work at a place, and they will only work at a place where they know they're going to have an organization that can be a good steward of their innovation and a good steward of what they've put together and have maximum impact. Revenue is not our primary driver, but it is important. And if you're able to generate revenue, you're able to do some really neat things as far as fostering more commercialization and more translation. So revenue is certainly a driver, and it's something that, you know, we take very seriously when we get an opportunity to reinvest that. Economic development. Places like The Ohio State University and Nationwide Children's Hospital can have a huge impact on the economy here in Columbus and here in Ohio by the activity that we undertake and the startups that we launch and the things that we support. The one thing my team would tell you is that we have a bias towards commercializing in Ohio, but it's not requisite. There are certain technologies that just don't fit here, and they're a fit in San Antonio, Texas, and that's where they're going to wind up. Our primary goal is always going to be to see the technology translated, but if it can happen in Ohio, that's wonderful. And then just public utilization. At the end of the day, that's our mission. We want to see these things utilized. We want to see them have an impact. So how do we get all that done, and how do we deliver, I guess, on this charge from the institution? And I think for me and my team, I hope would agree with me on this, that the culture we try to put forward to our internal customers and to the faculty is that it's all about the people and it's all about relationships. And so before I go into the process slide or anything else, I want to put our team up here. We have a really strong group of people in the OTC, and we really have kind of two tracks of professionals in our office. The first group, which we would refer to as kind of the licensing professionals, going across the top and then including Jocelyn on the second row. These are all people with advanced technical degrees. They're people who are able to dig into the science and really understand the innovations that are coming from our investigators. They build the relationships with those faculty, and they also build the relationships with external customers. The other half we have are people who have really started to specialize in a particular type of operations function within the office. So we have patent coordinators, we have people who are agreements coordinators, people who specialize in government reporting and compliance. And these are all things that enable us to make sure we're delivering a quality product. Our process is actually nothing revolutionary when it comes to tech transfer, and we've tried as many times as we can to make this graphic a little sexier, and it's just not. But the process is a lot of fun, and it's all about relationships. And really, when I put this up here, you know, the idea would be, you think about those people that I just showed on that last screen. When you engage with our office, you're going to be engaging with them, and you're going to be having them learn your science, having them understand the opportunity, and having them work with you through this process of invention assessment, understanding what a strategy would be, building the right deal for your technology. We are not contract wonks. We don't just have a canned recipe that we apply to every situation. We do believe every deal we do is a little different, and we have to have templates, and that's good to be efficient. The templates aren't bad. Those are good things. But we pride ourselves in the fact that we really work hard to understand a deal and understand an opportunity, and that we've been flexible on certain deals to apply maybe deal terms that wouldn't always look like they were the right thing to do, but they were for that situation. So the people are really important to this process. And then the other part I want to point out here, I won't read each box to you, but this is an important part of our process too, this whole not viable and feedback part. Part of what we're doing with our internal customers is building a relationship so that they, even when they don't have a commercially viable shot on goal with us, they're getting the experience of understanding why. And they're getting the experience of at least going through the process. And if we walk through this part of this overall graph, we're more likely to get another shot on goal with that particular faculty. Next I'm going to show some metrics for us just to, I guess, hopefully convince you the office is growing and we're working towards trying to drive our numbers up. It started to level off, I think, here in the disclosure numbers. If you go back to maybe 8-9 years ago, we were right around 20 disclosures a year, and now I think we're pushing 100, but until we develop either a few more robust resources to dig into the clinical side of the hospital further than we already have. We do work with them tremendously often, but not as much as we, I suppose, could. There's a lot of people over there. Or until we see additional faculty be recruited. I don't know that this number just shy of 100 is too far off of where the probably proper number of disclosures are for us. We could generate 200 disclosures this year if you wanted us to, but we wouldn't because it wouldn't be worthwhile and they probably wouldn't be quality. This seems to be the right number for us. The patents issued we've seen continue to climb. However, there's a particular portfolio that was filed back in the 2004, 5, 6 time range that's out licensed to a licensee and had several species variants on this particular, and that company in these 2 years just went gangbusters, filing patent after patent after patent with different species for this. So kind of artificially inflated those two. The number that I think is most important that I can share with you today is right here, number of deals. The thing we really, really care about is shots on goal. And so we've been driving every year to try and maximize the number of deals we're getting done. I looked at the team's numbers this past year with 84 disclosures and 29, I guess, not 30 deals done. I'm really happy with that percentage. It doesn't mean there aren't more deals we could have done, and it doesn't mean there aren't more disclosures we could get, but we're at a place now where I feel like, at least compared to tech transfer standards, we're We're doing an efficient job with that. And then we have been blessed that a couple of our technologies in the gene therapy space just lined up really at the right time with the market, and I think our team did a nice job of putting a good deal out there, but just as, if not the most important thing, our institution really had built an infrastructure that was primed and ready to go when the gene therapy opportunity occurred. And so we've seen a few successes in gene therapy that have given us some nice revenue and returns. The thing I would point out about here, and you can't see it on this graph, but I think it's important to note, in 2017, that was really one deal. I mean, of the 36 million there, all of it, like 35.5 million of it was from one deal. The rest were just, deal sheet that we were building. If you come out here to 2019, the year that we just finished, this, 30-some million is built on the back of multiple deals. And so this isn't a one-trick pony. These are a lot of the deal sheet we put together delivering. We're really excited about that. We've launched, I think, a total of 15 or 16 startup companies kind of during this team's tenure in the office. These are a list of the ones that are still active and alive. I guess if they've been acquired, they're not active anymore, but we're leaving them up there because they didn't atrophy like some of the other ones we've launched have. But I do want to point out there's some gene therapy names up there that you may recognize. Avexis and MyoNexus have had quite a bit of play in the news. But actually, there's a lot of opportunities we have here that aren't gene therapy where we're delivering some neat things from Scioto Biosciences, which is a prebiotic formulation that we think is really, really interesting in the gut-brain axis. Deep Lens is a company locally that's just created several new jobs with funding around applying artificial intelligence to pathology, and you're going to hear more about some of these other programs. Dr. Breuer is involved with LIST today. I don't know how much he's going to talk LIST or other things, but I won't steal any of his thunder. That's an exciting company that we have that we're moving forward. And then just to give you an idea on the economic development side of things, like I said, most of these, if we can keep it in Ohio, we do. But there are times where it doesn't make sense, and we got to take it elsewhere. And we'll always lean towards what makes the most sense for getting the technology translated. Just one or two more slides here. There are some things that we do to try to make the commercialization process easier and try and help bridge the gap between kind of the early stage discovery work that's traditional research and the place where we're actually palatable to a VC or to a potential licensee. One thing we've had a lot of success in working with is what we call the Technology Development Fund. There's nothing revolutionary about this. It's just traditional pre-licensed gap money. And so basically when you're an inventor that's come to us and you have some sort of experiment that will create a value inflection point, and we really do intentionally make it that broad. Because sometimes people say, wait a second, is this preclinical, or is this prototyping, or is it all of the above? The idea is if an inventor can come to us and say, hey, I can describe to you a way that I can use $75,000, and this is the data we'll get, and these are the reasons we know company XYZ wants to see that data, then we consider them a good candidate. We've used it for showing that the effect of a drug was sustained, down to prototyping a trait collar. I mean, we've used it for all different kinds of things, and we've had a lot of luck with creating license opportunities out of this. We've also used this technology development fund for pre-licensed technologies to kind of be the matching money for state programs that are targeted at the same development stage of technology. We did 3 years ago launch a life science seed fund with the state of Ohio managed by Rev 1, and we had some nice experience with that as far as it catalyzing investment from other seed funding sources. We've had a couple of companies that have given us a nice return on that, and we're excited about the future of those companies. And in fact, we felt this fund worked so well that we asked our friends at Ohio State University if they would like to maybe work with us to make an even bigger seed fund an opportunity. And we were all able to partner, put that together, and we have the Catalyst Fund 2 now, which is also in partnership with Rev 1, but very exciting that OSU gave us the opportunity to get in on a bigger fund like that. And then finally, like I said, referenced earlier, I think infrastructure is really important for what we do. We have a GMP facility for manufacturing drugs. We have the drug and device development services, which Chris is going to talk to you about a little bit later today, and really robust clinical research services. We are kind of focused, we're a hospital, so we don't get a lot of disclosures with circuit diagrams. We don't get, we get a lot of IT, and we get a lot of drug plays and a lot of just different therapeutic approaches. So That is us in a nutshell. I think that the most important part I would want to leave with you as a take-home is, we really understand in the tech commercialization office that we're not the dog, we're the tail. But we think if the tail's doing its job well, it's a happier dog. And it's a stretch. I'm working on that analogy. But the point I want to make is, no, I think that when you have a well-oiled commercialization operation that's generating returns, there's an opportunity to take that and reinvest it into the research enterprise and grow the research enterprise. And it's been a fun thing for us to get to live some of that recently, and we're hoping to live more of it in the future. So, any questions before Dr. Breuer comes up? Yeah.
What's the relationship with your OTC and OSU's technology?
It's a good, I'm right there, but it would have said good stuff anyway. It's actually a very good one.
What I meant is how many deals have been made that are returning dollars to both institutions?
So there's a memorandum of understanding that exists between the institutions, and so we really see that happen on two different fronts. So there's one front where there may be a technology that's just exclusively being managed by Nationwide Children's Hospital because all of the faculty that are part of that invention, as defined by the Memorandum of Understanding, are NCH faculty, even though they have joint appointments. And in those situations, because of the close relationship and some of the ties that exist between us, when revenue is generated from that, there's a formula by which portions of that are given back to OSU in recognition of that relationship. But then there's also several instances where, because of the proximity and the collaborations and the co-projects, we have a number of technologies that were co-invented by NCH inventors and OSU inventors. And in those cases, Kevin's team and our team work to negotiate an interinstitutional agreement, just like any other institution we would work with. And there's a prescribed revenue sharing, usually at a higher order of magnitude than what you would see in the others. So yeah, there's all, it's a well thought out process, I think, and it's good. Any other questions? All right. Without further ado, and he did give me permission to not use all of his big fancy titles because he's got a lot of them. The director of our Center for Regenerative Medicine, a good friend of mine and a wonderful inventor to work with, Dr. Chris Breuer.
Yeah, I can invent things, but I can't use a computer. All right, good. Thanks a lot, Matt, and thanks to the organizers for the opportunity to share some of my research today. This is a real pleasure. And thank you for all of you in the audience for coming. It's a dreary Monday morning, and you got up very early. I'm appreciative of your willingness to be here. So I entitled my talk Accidental Tourist, because in many ways, when I look at my career, that's what I feel like. I've been impressed over the course of my life how much chance and serendipity has to do with where we wind up and what we do, which is kind of surprising given where I come from. I'm a person that has always known exactly what they want to do. I was born to be a doctor. It was my fate. I never considered another career. And throughout my life, my path forward has been very straightforward. I took the steps to become a physician and marched through and did it. And I could never understand growing up why my friends had all this angst and anxiety trying to decide what they wanted to do with their careers. That was until I started medical school. And after completing my first anatomy test, I went to a keg party and had this profound realization that I had to do something after I was done with my training. Now, after 23 years of a journey, I think I might have thought far enough ahead to consider this obvious question, but I hadn't. And so I started to appreciate all the angst that my friends had been experiencing. Ultimately, I decided that I wanted to pursue a career in pediatric surgery, which entailed that you needed to complete training in adult general surgery and then go on and do a fellowship in pediatrics. And things worked out quite well. Once again, I was on a little bit of a treadmill and very comfortable working through the residency until a couple of the senior residents that were in front of me applied for positions in pediatric surgery and failed to match. And when I spoke to them, they explained to me that it was an unwritten prerequisite that if you wanted to be a pediatric surgeon, you had to take off some time and go into the laboratory. So I approached my program director and asked for permission to take a research sabbatical, and it was granted. So in essence, I went into the laboratory for all the wrong reasons. I had no innate interest in research. It was simply a means to an end. I was fortunate to meet a man named Jay Vacanti, who's a pediatric surgeon working at Boston Children's Hospital, and obtained a position working in his laboratory. He and a gentleman named Robert Langer, a material scientist from MIT, had developed this new discipline that they called tissue engineering. And tissue engineering attempts to leverage the cell's innate capacity to multiply and then self-organize into functional units that are working together. And they had developed a method using a device that they called a scaffold that would provide sites for cell attachment and space for tissue formation, and could be designed to serve as a template to control that tissue formation. And then the resulting tissues could be used to either repair or replace tissues that were either diseased, damaged, or congenitally absent. And this really would enable surgeons to do much more and have a much more profound effect. Vacanti and Langer were real early adapters in the notion of patenting and then working with venture capitalists to fund their laboratory. Dr. Langer right now actually has the most patents of anybody. He recently surpassed Thomas Edison. And at the time, they had a tissue engineering patent, a new matter patent, that they had managed to license to a company called Advanced Tissue Sciences for $150 million. And this was back when $150 million was a lot of money. And they were, in return for this licensing agreement, they would provide the research to advance tissue sciences research and development. And it was a real exciting time to work in the lab. The lab seemed to have the ability to blur the boundaries between science and science fiction. We had the dubious distinction of having as many articles written about us as we had articles coming out of the laboratory. And we graced the covers of magazines ranging from Science to the National Enquirer. And the group of people was really tremendous. They've all gone on to have interesting careers in academia, within medicine, engineering, and within industry. And it was a wonderful time in life, with one small problem. I was a rotten scientist. At the end of my first year, I didn't have a shred of publishable data. I hadn't done anything well. My intentions were great, but I just really didn't know what I was doing. I had inherited a project trying to create tissue-engineered intestine by putting intestinal cells onto a matrix and then trying to get this to transform into intestinal tissue. And I wanted to do this because as a clinician, I was dealing with patients with what's called short bowel syndrome on a daily basis. And I'd seen a number of patients die from this really devastating disease. And not only do we lose the patients, but you watch whole families fall apart. But I simply didn't have the research acumen to perform this sort of research. So I was forced to acknowledge my failure and really look in the mirror. And what I decided was that this was really kind of a naive project. At the time, we had no idea how to create a capillary network. We had no idea how to innervate tissue, both of which are prerequisites for creating functional intestine. We didn't really grow the cells very well, and our scaffolds were really not very sophisticated. So I decided to go back to the drawing board and look for alternative projects. And I was fortunate at the time that Dr. Vacanti had lunch with another surgeon at Boston Children's Hospital named John Meyer. And John was a congenital heart surgeon, and he spoke with Jay and explained how there was a great need to develop tissues for use in congenital heart surgery. Things like vascular patches, replacement heart valves, or vascular conduits were really needed in the pediatric population, where children could have life-saving operations only to outgrow them. And more importantly, these materials didn't require a capillary network or didn't require to be innervated in order to function. So the next year in the lab, I picked up this project and things went much better. In less than a year, we had a functional tissue engineered heart valve. We created the prototype for the vascular conduit, and it was really, really quite wonderful. And one of the main reasons why I was successful this year wasn't only because the project was good, but because Dr. Meyer introduced me to Toshi Shinoka. And Toshi's a congenital heart surgeon from Japan who was working in Dr. Meyer's lab. And for the last 25 years, we've had a wonderful collaboration. And in most collaborations like this, the reason why they're successful is because people bring in complementary expertises. And Toshi is this really hardworking, really intelligent person. He's highly organized. He's a very creative scientist. And I still haven't been able to figure out what I bring to the collaboration, but I'm happy to be part of it. I went back and completed my clinical training and then had to go into the military. And over the course of 8 years, sort of finished all this and was trying to decide what I wanted to do with the rest of my life. Now, normally within the field of academic research, if you've taken an 8-year hiatus, your career is over because what you studied 8 years ago is ancient history. But I was lucky enough that tissue engineering in our laboratory, we were far enough ahead of the curve that the rest of the world was finally catching up. Tissue engineering had matured into more of a science than it was when we were working in the field. And I'm in the military, and I pick up a copy of the New England Journal of Medicine, and Toshi's on the cover. And he had returned to Tokyo Women's Hospital. He had become a surgeon scientist, and he had translated the technology that we had developed together to the clinic. And it was really extraordinary to read about this. Toshi had partnered with a company called Gunze, a Japanese textile manufacturer, who had helped him improve the design of the scaffold and created a method for fabricating the scaffold that would enable him to implant these in patients. And for me, reading about this and contacting Toshi and congratulating him was really an epiphany. And what I realized is that what I really wanted to do was translational research. I absolutely love the honor and privilege of being a surgeon. There's nothing better than taking care of a patient and helping them get better. That's worth spending a lifetime doing. But I'm equally frustrated and get equally weary with those patients that come to you with problems and you can't solve them, especially when they come in with the same problems again and again. So being a surgeon scientist and being involved with translational research enabled me not only to help the patients, but also work to advance the science of surgery and hopefully improve our ability to take care of patients. So for my next step, I was looking for a place to work, and Yale University had started a thing called the Tissue Engineering Initiative. And they did something that was really very unusual in the field of tissue engineering. They focused. Most tissue engineering laboratories across the country fell into what I would call the short attention span theater. You were working on a million different tissues in a million different places. But Yale had decided to not only focus, but hyper-focus. Everybody that they recruited were making vasculature using tissue engineering. And it was exactly what the science needed at the time. So I set out to become a physician scientist. I worked with the NIH, was able to obtain funding and able to start a lab. And the key to my work is that what I really wanted to do was understand the cellular molecular mechanisms by which neotissue forms, because I thought, armed with that information, we could rationally design improved products. And so I wanted to take advantage of transgenics. I wanted to be able to use mouse models where we could turn on and off the different genes so we could understand these things on a much more molecular level. We developed methods for manufacturing our scaffolds on a much smaller scale so that they could be implanted in mice. And we developed microsurgical models that would enable us to replace blood vessels in the mice that were less than 1 millimeter in diameter. And over the course of 15 years, we were able to elucidate the mechanisms that govern neotissue formation. And based on our discoveries, we were able to rationally design strategies to improve our tissue-engineered vascular grafts performance. Over the same period of time, I was lucky to be able to recruit Toshi back to the United States and join me at Yale. And together we worked to bring tissue engineering to the clinic in the United States. Now, one of the most critical first steps in doing this was obtaining FDA approval. Toshi came to Yale in 2008. His prior study, which was performed at Tokyo Women's Hospital in Japan, was before the Japanese government regulated tissue engineering. So he only needed to obtain IRB approval in order to proceed with his clinical studies. So together we approached the FDA in 2008, and 3 years later we obtained approval for performing the first clinical trial in the US evaluating the use of tissue-engineered vascular grafts in patients. Now, to put the work in perspective, this was a 3,000-page document that gave us permission to implant 6 patients. Toshi's original IRB was a little under 12 pages. We selected to use the tissue-engineered vascular graft to treat children with congenital heart disease, so we could take advantage and evaluate the graft's most unique property, and that's its growth capacity. And in 2011, we performed the first implantation. This was our first patient, Angela, who fortunately did very well. Angela is now 8 years after surgery, and serial evaluation of her grafts using MRI demonstrate that, in fact, the tissue-engineered vascular grafts do grow, making them the first grafts with first man-made grafts with growth capacity. Around the same time, Toshi and I began to think about moving to other institutions. Yale's a tremendous place for performing basic science research, but it's lacking a bit when it comes to translating research simply because they're a fairly small institution. So we had a number of awesome opportunities, but really focused in on Nationwide Children's Hospital for 3 reasons. One was that they had very large, highly innovative clinical programs, which could serve as great beds for recruiting patients into our clinical trial. 2, they had phenomenal infrastructure, beautiful buildings, and great capacity for doing GMP sort of work that's required to make products for implantation into people, in addition to incredible large animal facilities and small animal facilities for performing our research. And probably most importantly, their leadership had a real desire to perform translational research because they felt it was an essential part of their mission. In addition, there were a number of incredible programs that really accelerated our research, and I met 2 individuals that have really had a profound impact on my work. The first is the wooden squirrel, and the second is the wooden rabbit. No, actually, Matt McFarland and Chris Schilling and their teams have been extraordinary, and I'd like to highlight some of the work that we've been able to do together. They're very enabling. In 2014, we implanted our first patient with a tissue-engineered vascular graft at Nationwide Children's Hospital. That's Anderson, seen here. Unfortunately, Anderson had a narrowing in this graft, as did the subsequent 2 patients. These patients required treatment with balloon angioplasty in order to fix the stenosis, but then went on to have uncomplicated courses beyond that. There were no additional graft complications, and all patients are doing well and have demonstrated evidence of graft growth 5 to 8 years after surgery. But in light of these complications, we decided to go back to the drawing board and returned to the lab and continued our work trying to understand why some tissue-engineered vascular grafts narrow, while other tissue-engineered vascular grafts go on to form stenosis. And one of the things we've been able to accomplish over the last 5 years, which has proven really, really valuable, is in collaboration with a group from Yale and Stanford, we've been able to create a computational model that can actually describe and predict neotissue formation in our tissue-engineered vascular grafts. Using this model, we ran simulations which accurately predicted the stenosis that we saw in our clinical trial, but surprisingly, predicted that the stenosis would have gotten better by itself. So this gave us a really interesting opportunity to validate and test the model. So we performed a large animal experiment which showed, in fact, that tissue-engineered vascular graft stenosis is reversible. Additionally, we continued with our work trying to develop methods for preventing stenosis based on understanding its mechanism of action. And what we discovered is that treatment with a generic drug called losartan for a short period of time immediately after implantation of the tissue-engineered vascular graft could also be used to prevent or reduce the incidence of stenosis. And so in collaboration with Kevin Bossi and Chris Schilling from the Drug and Device Development Services, we shared this information with the FDA and recently approved approval for the next phase of our tissue-engineered vascular graft study, approved for implantation of 24 additional patients with congenital heart surgery. I can't possibly begin to thank Kevin and Chris for their extraordinary work. They single-handedly really accelerated the process of translating this technology from the clinic, and they offer a really amazing service, and clearly are national leaders from a regulatory perspective, not only for the regenerative medicine space, but in the gene therapy space. Additionally, we've continued our work trying to bring tissue engineering to the clinic. One of the things that we've also developed is a closed system that you see drawn here. This closed system is simpler, safer, and less expensive than our current open method, which requires the use of a clean room, and therefore increases the utility of the tissue-engineered vascular graft and will make it readily available to other institutions, in fact, any institution where congenital heart surgery is performed. And I could not have done this without the help of Kyle Murray and Matt McFarland and the whole technology commercialization office. They're true and trusted partners and provide guidance and counsel in helping us to try and work with industry to bring our technologies to the clinic. And probably most excitingly in the laboratory, we've had a number of really interesting discoveries. And this was a perfect example, again, of chance in the laboratory. So in our quest to understand the molecular mechanisms of how stenosis forms, we originally used a chimeric model where we could take human cells, put them on a tissue-engineered vascular graft, and then implant that in an immunocompromised mouse model, and then study the fate of the human cells to understand what was going on. And we had this beautiful model where we'd implant these human tissue-engineered vascular grafts into these immunocompromised mice. And we really, really wanted to use it to study why stenosis forms in tissue-engineered vascular grafts, but the model wouldn't work. We couldn't get the model to stenose. And for 3 years, we kept going back to the drawing board and putting in these grafts and trying to get them to stenose so we could study stenosis, when finally the light bulb went off and we discovered this isn't the problem, this is the solution. This is what we're trying to achieve. And it turned out we were able to isolate the immunodeficiency to a defect in the LIS gene, the lysosomal trafficking regulator gene. And this was really exciting because this genetic defect was powerful enough to preclude the need for cell seeding. So we've gotten to the point where we can now tissue engineer with just biomaterials, which is really revolutionary and will enable us to continue to advance the field of tissue engineering. Together with Cameron Best, a medical student at Ohio State University, we continued to study the role of LIS gene in regenerative medicine and also found that it had a variety of other very important immunomodulatory effects. It controlled wound healing. It prevented scarring. It prevented adhesion formation. It's a powerful immunosuppressive that can even be used to enable xenotransplantation. And it's even important in fibrosing diseases such as liver fibrosis and pulmonary fibrosis. So we started a small startup company called LIS Therapeutics in an attempt to continue our research investigating the role of LIS. We've been able to obtain NIH funding through the SBIR and STTR programs. But more importantly, we've been able to receive substantial support from Revro Inventors. And I'm very appreciative to Tom Walker, Wayne Embry, Ryan Hillon, and others from Rev1 who've been so helpful. I'm also really appreciative of the extraordinary work of Meg Rowe, our CEO, and Miu Hu for helping us get the company started. Happy to announce that we're now one of Columbus's Fortune 500 companies. Actually, that's a bit of an exaggeration. Our headquarters is located just outside of Columbus. So perhaps the most famous accidental tourist is Forrest Gump. And I always love this story. Really the theme throughout Forrest Gump is what controls our destiny? Is it fate or is it chance? And at the end of the movie, Forrest concludes that it's neither fate nor chance, but both fate and chance working together. And I always found that to be intriguing. Tom Hanks said that really the theme of the movie was that how we respond to chance events in our lives will determine our destiny. And they used the theme of the feather as a symbol for this. And so my final parting words are to be careful and look for the feathers in your life. I've had a few pop up, and they've been very helpful to me. I'd like to thank the Ohio State University Center for Clinical and Translational Research, and I'd be happy to answer any questions. Thank you very much for your attention.
In the interest of time, could we hold off the questions till the very end? So, and then you could also use the mic because I've already gotten several emails saying that they can't hear the questions. So thank you. So is that okay? So we'll do the Q&A at the very end. Chris Schilling? Yes. Thank you.
I can do this. Aha. Good morning, everybody. Thank you for that talk, Dr. Breuer. I apologize for everyone. You just had a wonderful science talk from Dr. Breuer, and I have a guy up here who's going to talk about laws and regulations, so hope won't you put you to sleep here. I'm the director of the Drug and Device Development Service at Nationwide Children's Hospital. We're a program focused around regulatory affairs, and I don't have much here, so I'm going to just kind of fly through a couple slides here. You're probably going to get some time back, but just to talk about what we've done as a service for our organization as well as Ohio State. We are built within the Ohio State University as a service under the CCTS, so we're accessible through university, through the CCTS as a service. We offer voucher programs that help support our access to our service. But to tell you a little more what we do, we're a regulatory affairs office. So what is regulatory affairs? Regulatory affairs is the units within companies, typically pharmaceutical companies, that are engaging with your regulators. In this case, for drugs and medical devices, it's the Food and Drug Administration that's regulating the access of those materials to the public or through clinical trials and the development of those products. And so we're a unit that liaises with the agency on the behalf of our institutions to talk with them, understand what their concerns and challenges are as we develop a technology to ensure that we can hit good benchmarks and good milestones to ensure success in getting that technology through its development stages, from preclinical, early safety studies, to actual engagement in clinical trials, seeing that we can do or meet what their expectations are in the development of this technology, and hopefully finding great success in a market opportunity. Partnering with our great colleagues in the OTC office is to find those good partnerships to find commercial opportunities. With the value we've generated on this technology through the engagement with the FDA, we should hopefully have success. So we liaise with the agency. We conduct interactions with them. These can be a variety of different formats, from meetings to actual filing of different requests, paper submissions, a lot of documents. Dr. Breuer came from Yale, and he described his story where, 3 years after first engagement, they finally got their trial moving. That's an extensive amount of time. This is not intuitively understood, the process it takes to get the FDA engagement and get the elements necessary to allow you to take a device that's been developed in an early stage into clinical use. We really try to realize on our resources and our experience to help you with that process and to diminish that time frame. So it's no longer 3 years, but it'll take time, and the paperwork is extensive, but we've got the right people in place to help support that and move that along in a more efficient manner. We also touch upon unique things that maybe not everyone understands, is these opportunities for designations. These are other feathers in the cap that we can pursue and gain that generates additional value maybe to the technology, depending on its use. If it's orphan, if it meets a unmet medical need and a rare patient population, the agency provides a lot of benefits to those technologies by getting designations awarded to them through the orphan drug or humanitarian use device. And Dr. Brewer's tissue engineering graph came to us with that designation already in place, but those are great benefits the agency may offer you to help expedite some of the development and grant you additional interactions with them to support its development. And then as our office works in collaboration with our investigators, is we do support these for life, through the life cycle of development, maintenance, and continuing reporting to the agency, as there's a lot of responsibilities that we take on when we're doing this kind of work that's engaged in the human trials. Our experience is predominantly in the biologic space. We've come to understand a lot of different elements to launching technologies that are based on biologics, and we are in a stage of medical development that's unique now. There's an abundance of new biological opportunities, and based on the development of the human genome and understanding of that, we're really grasping how many opportunities there are out there. The development of biologics as a therapeutic is much more complex than we've ever known in the prior medical field. A biologic technology has an abundance of critical elements that makes it complicated to both manufacture and to maintain stability. So we work very hard to understand these complex cases and work with the investigators to help ensure that we're getting the right data to support its development with the agency. We kind of see ourselves as a little bit of a balancing act between both the commercialization side and the research. We're interacting with both parties. We sometimes see ourselves as also a good steward of data with our commercial partners. When the OTC finds a match, and we're still developing this technology in our institutions, we work with that partner to ensure we're getting the right information to them in a timely manner and ensuring it's good quality and supports their needs. And fundamentally, this is just a concept that I think a lot of people already get, but just to reiterate that we find ourselves uniquely in a position to help where we have great discoveries going on in the academic space. And I think the OTC offices have shared to us plenty of times how you can see these great discoveries occur, but to get it to that beautiful spot in the green pasture where it's marketable technology takes a big leap over this valley we always refer to as the translational valley of death. How can I take this early discovery in a lab and get it on the right track to support its opportunity for value with a commercial partner? And we really try to build that bridge through hitting certain benchmarks and milestones in its development pathway. And this is basically our formula for doing some of this work. This is based really on a biological scale, but can be applied in a number of other technologies. But our engagement, really earlier the better. If you have an idea and you're just now getting endpoints that show that I have a biological effect that could impact a disease or population, look at it as a proof of concept. Bring that to us. Let's talk about that. Let's see what that value is. And we can put our team in places to start evaluating what are the gaps in that technology. What are our concerns? We put our FDA hat and say, how would they poke a hole at this? What are their challenges in saying, have you done enough to support that this is an efficacy outpoint? Or is there a safety concern we're not looking at yet? Based on some of our gap assessments, we can start encouraging you, hey, we might be at the right stage, and just go ahead and have a conversation with the agency. The most value you can get is by having a dialogue with them and getting feedback directly from those who are going to make a decision about whether this product can be in a clinical trial is through those meetings. And those interactions, believe it or not, are actually great value incentives to a commercial partner. If you now can sit with a commercial party and say, hey, I have a technology, I've generated an outcome, and yeah, actually I talked to the FDA about it, and they've given me the direct feedback that I'm on the right path, but I got to do X, Y, and Z. Wow, that's an impactful value that you've now just shared with a partner that says, you know what you're doing, you've got a pathway, you've got actually a plan that shows I can continue to march along this pathway and find probably a good chance of success. So we evaluate opportunities for engagement. In the biological space, we have various meetings. The agency right now is very much been, or historically has been, very supportive, working with academic organizations like our own to say, hey, we know there's great ideas, great technologies coming from you. We would like to be able to be a part of that and have a conversation. And the biologic space is booming right now. There's a lot more in gene therapy and other forms of technology that are really impacting the field. We sense a little bit of that engagement maybe dwindling a little bit, but they're still there, and we're still engaging them as much as we can to ensure we got good feedback. But I think the important aspect here to see as we go through this pipeline and going through different interactions with the agency and then taking in on opportunities for either manufacturing and critical studies, based on the feedback we've had from the agency, our value for investing partners continues to rise. And so I think that's an important philosophy that I think our OTC offices and others have encouraged, is that we may allow an opportunity to us to hold on to that technology a little longer, maybe not license it at the first deal, and just to be able to generate more value through this engagement and kind of incubating the technology and generating more value in it as we've continued to develop it internally at our own institution. And to really understand that we have a number of programs that we've held and maintained regulatory responsibility with through our office all the way out to the onset of initial first patient dose. And that's kind of uncommon. And just to show you just some of the impact we're trying to have, this is our gene therapy pipeline. I know Dr. Breuer, I've got more material on him, but just to show you just some of the areas we're working on heavily is various programs with different indications with our focus is being able to hit what can we do in designations. These are orphan and rare pediatric disease designations the agencies are granting us that generates value. You also have designations that generate expeditious reviews, including breakthrough, regenerative medicine and alternative tissue fast-track designations, a variety of other opportunities that we're helping to move these programs forward with. And so, again, we will engage at a very early preclinical stage to help support the development through, my laser just died, through the various steps into early phase trials and support that we can see these technologies continue to grow until that there's finally an opportunity where the technology may be licensed to an external party and we find become a cheerleader on the sideline. Once that technology is transferred, regulatory FDA interactions are then pushed out to that company. We support them as much as we can. We become a cheerleader for that technology to see it go through into commercial, hopeful opportunities. Again, here's some of our opportunities in other cell and viral therapy, and we're very excited to see the successes with Dr. Breuer. I don't see Kevin Bossi with us, but he's been integral to helping that program, as Dr. Breuer mentioned. And I think I just wanted to just highlight the team. This does take a team, and this is the drug device development team. Kevin's been integral to the tissue engineered vascular graft program. Let me cut my slide out. Bring my crew back? There they are. A number of them are probably actually watching us right now from Nationwide Children's. So, Rachel's here with us. She's been our regulatory specialist on the gene therapy portfolio. We also have Sue Marting, oversees a lot of our off-label use. We have our new recruit, Neil Patel from Ohio State, who's doing his master's program in regulatory affairs, as well as our administrative assistant, Christine Cromaldi, who, thank you, Tanya and Christina, working so hard to bring this venue together. Appreciate the time, and I'll be happy to answer any questions.
So we welcome questions from any of you for any of the speakers, starting with Scott, Jan, Dr. Breuer, as well as Chris, and definitely Matt McFarland. So if you have a question, please come up to the mic and ask your question so that our colleagues who are listening through live streaming at Nationwide Children's Hospital, as well as on their own devices, can hear the question that was asked. Thank you, everyone.
Thank you all. Chris, what is your experience with combination device and drugs?
There you go.
Shilling.
Hey, Carson. How are you? What's our experience with combination drugs and devices? Yeah, so actually the story you heard from Dr. Breuer is actually a combination product. Both a device, as you see the scaffold he's utilizing is a biodegradable scaffold that is regulated as a device, and then the biologic matrix is the bone marrow mononuclear cells that are derived from patients then are seated on that, and that's the biologic side. So that is actually regulated as a combination product, which is very unique. It's actually a really exciting product where you have a variety of different groups within the agency combined to evaluate it. You have CDRH, your Center for Device and Radiological Health, that oversees all your med devices, all your pacemakers and so forth. They're the ones who oversee those devices. They're at the table because they're looking at that scaffold. And then we have CBER, the Center for Biological Evaluations and Research, which is the Biologics office that oversees like your cell therapies, your viral therapies at the table too. And for somewhere along the line, they flipped a coin and decided who was going to take the lead on it. And right now, it's CBER, the Biologics office, is evaluating or taking the lead evaluation role, but it's regulated as a device. So it has an investigative device exemption. So if you live in this space, it's just a unique scenario where the agency is kind of working collaboratively together to oversee that, and it has some nuances to it that make it a little more challenging to wind our way through the pathway. But that's one good example. We don't have many of those. Those are pretty rare. We have a small smattering of devices we're working with in mostly non-significant risk scenarios where they're not life-supporting medical devices or significantly diagnostic use. And then we have an abundance of biologics right now. Answer your question?
Is it on? No? Nope. No. Hello? Okay. Question for Chris Breuer. So lots of changes recently in how cells are thought about in terms of their activities in regenerative medicine. So the idea of stem cell is evolving tremendously. The idea of anti-inflammatory cells and drugs is evolving tremendously. I wanted to get a really, that you could probably talk for an hour about that, but wanted to get a really focused question on whether you think the industry is ready to provide cells for therapies for these different roles that we're now discovering different types of cells have. We don't have uniform cells anymore.
Sure, thanks for that question. I do believe that industry's ready, but one of the things that will enable them to be ready is understanding mechanism of action. So if you understand how your cell works, it'll obviously make it a more straightforward path to get through the regulatory pathway. And more importantly, if you try your product and it doesn't work the way you anticipate, you're more likely to be able to redesign it so that you can ultimately get it to work. So I think having a firm foundation in understanding mechanism of action of your cell product is really critical.
Hi, good morning. I don't. This is probably for the group in general. I'm not sure who's the most qualified to answer the question. What's your experience with therapeutics in pregnancy and repurposing some medications to be used in high-risk pregnant conditions? There's not a lot of devices in pregnancy, but there's a lot of interest in. I mean, over the past 21 years, there's only 3 drugs have been approved in pregnancy, and there's a lot of medications that we use outside that can potentially be used to treat a lot of pregnancy-specific conditions. So, thank you.
Sure. I don't have any experience with pregnancy, but we have forayed into doing fetal interventions, which has to do with pregnancy. And the first challenge you run into is anytime you do a procedure with a mom and a fetus, you've got a potential for a 200% mortality. So the regulatory bar is going to be higher. That said, all drugs and devices need to be tested for teratogenicity and toxicity. So I think a lot goes into that. I think at the end of the day, the challenges you'll run into will have to do with the potential impact of your product. So if you're dealing with a disease where there's 100% mortality, then the cost-benefit ratio is such that you'll get through with less regulatory burden. While if you're dealing with something that's more of an incremental improvement, the bar gets higher and higher.
Please don't sit down.
Just one thing I wanted to add to that too. I do think there's been a bit of a paradigm shift in drug discovery, in that I think when I was in pharmacy school 20 years ago, the concept of addressing a market of that size was just kind of silly. It wouldn't have happened. And I think with technologies like gene therapy and personalized medicine and curative medicine, we've seen the for-profit sector start to give an honest look at these really super rare populations with devastating outcomes. And so it's probably a little different game than it was 15 years ago. So, sorry.
Oh, of course.
Sorry. Yeah, I mean, this is great. I appreciate the input. But I have an IND for a drug status to be used in high-risk pregnancy condition, and it's been a whole lot of delay by the FDA. We have a lot of data on it, et cetera, to treat specific pregnant conditions. But then moving it to the next phase, it seems there's a lot of like disconnect and there's a lot of scare in treating pregnant women. It's an orphan population that's really not studied. So maybe we can take this offline later on to see what would be the next step to move this to the next level.
I can say our office has not had much experience with anything specific to that, but we are aware of the challenges and some of the questions that would be asked, and particularly also what may be required from a data perspective, and be happy to talk to you, and I'll give you my card.
Well, I have several questions and comments. First of all, 3 years for FDA approval sounds daunting, but how far outside is that from a normal progression? That's one sort of comment and question. It didn't seem unusual to me, and how quickly is that being accelerated here and at OSU?
Sure, great question. So I think it was slow compared to industry standards, and I take responsibility for that. You know, Toshi and I went to speak to the FDA and had no industry background. We were two, you know, surgeons, and we had some research experience. We had something that we thought had value, and we just spoke with them honestly, and they were very kind to us. I think they had a soft spot for, you know, academicians trying to do something without industry support, and so they really helped us through the process. At the end of the day, I'm a big fan of the FDA. I think you need that sort of governor on you. You need somebody to curb your enthusiasm. You need to make sure that you're thinking about things from all different perspectives. And so I would guess that, you know, the sort of work that we did for a more proven pathway is something that industry could do in a matter of months. And I think with Chris Schilling's group, we can meet or exceed those standards now, which is really unusual considering how novel a lot of this stuff is. I think the more experience you get in an area, the better you get at it. And so I think there's a really incredible opportunities to leverage what we have at Nationwide and Ohio State University to learn from each other's experiences, because I think these sort of services are invaluable.
So now to some scientific question, maybe that your first patient for engraftment and others evidently did not have the stenosis. So I'm sure you've done this, but what's the correlation between list and not having stenosis? What are those correlations?
Yeah, another great story in not having your eyes open. So when we went into the laboratory, the notion was that tissue engineers were engineers. You know, the cells were the bricks and the scaffold was the framework upon which you built things. And so I went in with this preconceived notion that that was the truth. And when we finally started to do the background science and did the cell tracking experiments, I had this poor medical student named Jason Rowe who repeated this experiment 3 different ways, and I wouldn't believe it because it was not consistent with my preconceived notions. And he kept coming back and saying, the cells disappear, the cells disappear, the cells disappear. And what we discovered was that the way the tissue forms is immune mediated. What the cells actually do in my type of vascular graft is they serve as an immunomodulator. And it turns out that the cells that are most critical in the tissue formation are our own inflammatory cells that infiltrate the scaffold as part of the foreign body reaction. So it's actually the macrophages that are critical. Mm-hmm. If we treat animals with, you know, macrophage, you know, poisons, or if we use transgenic animals where we can conditionally remove the macrophages, not only do you not get stenosis, but you don't get tissue formation. So it's actually the key to the whole thing. So in our particular type of tissue-engineered vascular graft, this sort of got turned into a wound healing experiment, which has been really good to work with.
So I'm glad that medical student in his failure didn't lead him to go elsewhere. So you mentioned losartan, mm-hmm. in fact. So
yeah,
could this be a repurposed use that could be patentable?
Yeah.
So sort of my first question. And second, is this immune modulatory? I've never heard of an ARB being.
Yeah, so it's interesting. What we have, I've got this incredible microsurgery core. One microsurgeon in particular, a guy named Tai Yi, who's sort of like the Yoda of the tissue engineering world. And Tai can do more things in mice than most people can do in any other species. And so if propose it, Ty finds a way to do it. So one of the things, he created a model where we would remove the spleens from our animals, implant a tissue-engineered vesica graft, and then implant a GFP spleen, a spleen where all the cells were green. And then you could track where the cells were going. And what we discovered is that our grafts were filled with green cells. So the spleen turned out to be a really critical source of the monocytes and macrophages that control the tissue formation. And it turns out that losartan modulates the release of cells from the spleen. So it's a way of altering the immune system to improve tissue formation. And we were able to patent it. And it's part of the next phase of the clinical trial, so all the children will receive losartan immediately after implantation in an attempt to improve graft performance.
Great. I forgot my last question, but those were terrific answers. Thank you.
I think Chris had a comment.
I think it was just referring back to your first question. Actually, I'm forgetting what that was. I was going to just a comment on just on the regulatory pathways are still developing, but we can talk more offline.
This is for Chris Breuer. The phrase in your presentation that most resonated with me, and the one I'll take away, is that you went into the lab for all the wrong reasons. And I got to thinking about the fact that you were passionate about a particular population, drove your research questions and drove your research directions. How do we get that story more in front of our medical students now? You know, the students who think they just want to go down the clinical path and don't realize that what they're carrying with them is a passion that could turn into incredibly valuable discoveries.
Yeah, and that's what I meant by accidental tourist. All of a sudden, I'm doing research, I'm working with the FDA, I'm starting companies, so you never know where you're going to wind up. I think the key for me was, just having a goal, and my goal wasn't really to be a doctor. My goal was to help people, and that I thought that there was meaning in life if I could do something that could help people that were sick, and that would be a life worth living. And so I think just having the big picture is probably important. And then secondly, I would say. My lab's awesome. My lab's my passion, and the people in my lab are extraordinary. And the people that tend to do the best in my laboratory are the people that still have that childlike enthusiasm and belief that we can do what we set out to do. And that was the whole fun part about being in the lab the first time, that it was okay to dream. Not only was it okay, it was essential. And if you're going to dream, don't have small dreams, have big dreams. Go for it. So I think that there's I think there already is a great future, and I can tell you from the people in my lab that are currently in my lab that we're in really good hands.
I remembered my question. So has anyone made progress on the short gut syndrome, which would be important because it would speak to whether your lack of success in the laboratory was specific to you or to the project?
Yeah, almost immediately. It was embarrassing how fast progress was made once I stopped working on the product. I wasn't very good.
I do remember what I was going to say. It was to your question about the timing and how long it took 3 years to get that program going, and has that changed at all? I mean, it really depends on the program. We can't just look in a crystal ball and predict it, but we are, at least our office is able to expedite things. It really goes to what does the field know and what is the science already established. In a lot of cases, we're trying to generate opportunities for therapies for things that don't exist already, and the burden is, do we have good models? We have to establish proof of concept, proof of efficacy in an early stage, and sometimes the models we utilize to generate that are not established, and that sometimes can delay things about getting buy-in. Do we have the right endpoints, and have we generated enough data to give evidence that we're making a clinical impact? Elements like that can push things back where you're still dealing with timeframes in the years sense. But if we have established models and we have established opportunities for generating clinical outcomes, The timeline can be shortened. We're still in the year or 2 years, but our office right now, particularly on good platforms, we're working like particularly in gene therapy where there's been strong platforms already established. We know what the pivotal IND enabling studies typically are designed for, how those are built. And we already have a clear picture of how we need to get there. We're able to really minimize those down to just the key elements about getting the work done and not, reinventing a wheel and trying to fumble through what are the right experiments. We can hit the marks pretty clearly on that.
I don't want to, we have some time built in for networking, but I don't want to stop the discussion if there's another question or two. Okay, great. Well, I'm Bruce Weinberg, and along with Tanya Matthew and Carolyn Crisafulli, I helped to organize this. Again, we are really thrilled to have everyone here, both physically in this room, at NCH, and on your devices for the people who are watching there. I want to thank Scott and Jan, obviously Chris Breuer, Matt McFarland, Chris Schilling for your presentations, all of you. It's really great to have you. Our sponsors, the Center for Clinical and Translational Science, OSU's Corporate Engagement, NCH's Office of Tech Commercialization, and then, of course, Tanya, who is truly the brains behind this operation, and Carolyn. I also want to put in a bit of a plug for our next presentation, which will be on March 16th. And that will be Melissa Bailey, and she will be highlighting Reach for Commercialization, and Mary Uhas, who will also talk about Reach from sort of her perspective. And I can say, I suspect that most people are familiar with Melissa and Mary, but I know and have heard them both, and they will be very dynamic speakers. So it will be worth getting up early on a cold March Monday morning to come see them, just as it was well worth waking up early on a cold February morning to hear the Chris's and Matt and Scott and Jan. So thank you very much. We have some time for networking, some more caffeine, empty carbs, go at it. Thank you so much for being here.